over the basics of the genetic code now. What do we need to know? So genetic code. The genetic code is unambiguous. So let me just point out, in the last video or our previous discussion, we were talking about how the genetic code was solved, how each codon, each of the 64 codons in the genetic code was linked up with an amino acid. We didn't really go over the stop codons, but we don't need to worry about that. This video, I just want to cover the important things to know about the genetic code moving forward. So the genetic code is unambiguous. What that means is if we have a codon like CUC, when I write a codon, I'm always putting the 5' end first, 3' end last. It specifies one and only one amino acid. In this case, it will be leucine. So all the codons specify one thing, one amino acid or a stop codon. The genetic code is also degenerate. What does that mean? So that means if we look at a bunch of codons like CUC, and I won't put the 5' and 3', CUU, CUA, I won't put those 5' and 3' for these guys, is that we can have, look at 1, 2, 3, 4, 5, all of these specify leucine. So more than one codon can specify the same amino acid. So it's unambiguous. Each codon specifies only one thing, an amino acid or a codon, or stop codon, sorry. But that's unambiguous. Degenerate means we can have different codons specifying the same amino acid. Now the genetic code has one start codon, and that is AUG, which codes for methionine. And sometimes there are some strange exceptions to this rule, but for the most part, most proteins are going to start with methionine because AUG is the start codon. Also, AUG can be found in the middle of a protein, right? So AUG is the start codon, but if you see an AUG in the middle of a messenger RNA sequence, well, that means there's a methionine needed in the middle of a protein sequence, in the middle of a chain of amino acids, or somewhere in the middle. It doesn't have to be in the middle. You know what I'm saying, I hope. Okay, stop codons. There are 3 stop codons, UAA, UAG, and UGA. These tell the ribosomes to stop making the protein, stop making the chain of amino acids. And I should say universal. So scientists believe that the genetic code evolved once and only once. And there are some exceptions. There are some obscure organisms, for instance, that maybe took one of the stop codons and changed it to coding for an amino acid. Those are very rare. For the most part, the genetic code is universal, seems to have evolved only one time, and all organisms share the same genetic code, with that very few exceptions that we won't need to worry about for this course. So what does that mean, the same genetic code? That means CUC, if that codes for leucine in humans, it also codes for leucine in fungi and bacteria and viruses. Okay, and 2 last things, we'll combine these: non-overlapping and comma-less. And these things seem obvious to us now, but when they were solving, the genetic code and trying to figure out how messenger RNA was used to make proteins, these were things they considered. And what does non-overlapping mean? So let's say this is the beginning of an RNA sequence that will be translated. Non-overlapping means this is a codon, and then this is the second codon, and this is the third codon. That's non-overlapping. Overlapping would say this is codon 1, this is codon 2, this is codon 3. So the ribosome would move one position at a time. That's not true. The genetic code is non-overlapping. Comma-less, what does that mean? So it also means the ribosome reads one codon and then it moves ahead to the next 3 nucleotides for the next codon, and then moves ahead to the next 3 nucleotides for the next codon, and so on and so forth. If it had commas, what that implies is that, let's say this AUG, this is the first codon, this would be a comma. Skipped. GAC, the next G, this would be a comma. That's skipped. Ribosome goes on to the next one. Or there could be 2 nucleotides as part of the comma. But that, we don't want to worry too much about that because that's not true. There's no punctuation marks, no commas. It is comma-less. So there's essentially no pauses, no skips. It takes 1 codon. It's non-overlapping, so it goes to the next 3 nucleotides for the next codon, next 3 nucleotides or the next codon, and that's how it translates, or that's how the genetic code works. Okay, so those are the important things about the genetic code. In the next videos, we are going to look at RNA polymerases and how they transcribe RNA. Okay, see you in the next video.